Hmm, it looks like this is not working the same. Can you guys hear me?
Yeah, I
Yes.
can hear you.
All right, I didn't get the button coming up saying start the audio, and this time it did. Last time it didn't. That's weird the way this is working. All right, let me share my screen here. And I saw that we're doing something different today. So let's go ahead and talk about the Unknown Project. The Unknown Project is something that you will be doing this term, and it'll be all online. It'll be where I'm going to give you a number, and it's going to represent an unknown bacteria. And you're going to try and figure out which bacteria the number is. Is that clear? So all of it's going to be done online. And
When does this start?
sorry,
When would this start?
it starts today. I haven't
Okay,
given
thank
you
you.
the number yet, but I'll try and get that out tonight after the lab. Anyways, that's that project. It'll be all done online. And some students get confused about it because the project initially was you would be going into the lab and I would give you a bacteria, and then you would run different tests on that bacteria to try and identify this species of bacteria. And we're going to do the same, except I'm giving you a number. And yes, you're going to do different tests, but it's all going to be done online. Is that clear? This project will be done all
Yes.
online, okay? And if you have trouble, meaning you're confused by it, send me an email so I can try and clarify your confusion. The first, well, I'll probably send you two emails. I'll send you one, hopefully tonight, giving you your unknown number. And then later, will that be a week later? I think it'll be a week later from tonight. I will give you your first results. And I'm going to choose your first 3 tests that will be run, and I'll go ahead and show you what the 3 tests are. Well, let's get into that later. I will send you 3 tests that I chose, and I'm sending it to everyone all at once. And the reason for doing this is to reduce the number of contacts between me and my students. We figured that each student would want to run something like 5 or 6 tests to identify their unknown bacteria. So I would be having 5 to 6 contacts with each student. And times 20, I just did the math and said, you know, that's just too much for one project. And so what I've done is I have chosen your first 3 tests for you, and I will just email that information to you in 1 week's time, hopefully. And that will reduce the number of contacts between you and I by 3. Am I sharing my screen?
Yes.
Yeah, you are.
Then you just saw that the lecture lesson is finalized, and I can now put that up on my YouTube site, but I'll do that sometime after the lab.
I was also gonna tell you not to forget to post the last lecture
You're
on
talking about
Canvas.
the
PowerPoint.
you're talking about the slideshow?
Yeah.
Okay. Yeah, I'll get that done as soon as I'm done talking about the unknown project. All right. So that's the unknown project. Let's talk about it in more detail, and we'll go over the project. You have the see if I can get this to shut. kind of. You can get the instructions that I'm going to talk about from, I think it's week 5 lab. And it's the unknown instructions. We'll talk briefly about the sample journal, building a dichotomous game plan, the unknown list and the unknown notes, and the grading rubric. I'll talk a little bit about all of these, but I'm going to spend most of my time talking about the unknown instructions. And hopefully you've looked that over and you will be ready to talk about this. And then on Thursday, we'll have the disc diffusion and antibiotics lab, real lab. But today I need to talk about the unknown project, and hopefully I'll get that emailed to you and your unknown number, and then we can start it. And you will not get the results, assuming I get everything to you tonight. You will not get the results until next Tuesday. Okay. And then you will look at those results, try and figure out what species are under consideration, and then you will determine your next test to run. Okay. So after a week's time, everything will be due to you choosing which test. Only initially am I choosing the first 3 tests. And like I said, the reason for doing that was I thought that this project had way too much interaction between me and the students for one project. And so I'm trying to decrease that by, I'll send not what is it called? Not 3 times 20. If there's 20 students, I don't know. Some students may have dropped now, but 3 times 20 would be 60. And I'm only going to send out 1 to everybody instead of 60 emails. And it just reduces the number of contacts. In reality, the first 2 emails will come from me as an email and Canvas. But after that, all of the contact will be between you and I will occur in the unknown notes page. And let me go ahead and open that and show you a little bit about it. It tells you how to do the unknown notes. I won't talk about that now, but you'll have a link up here for uploading a page. And each time you want to give me information or request a test, you will upload a page, and then I will look at it. And I promise to look at these pages at least every other day. And usually Monday through Friday, I'll look at them every day. But on the weekend, I'm not going to promise that I'll look at them every day. So if you turn in your notes on Friday, after I've already looked at the pages on Friday, I may not get to it. I may not get, well, I won't get to it for Friday for sure, but I may not get to it on Saturday. I may not get to it on Sunday. You may not get the results until Monday, meaning I'm not promising that I'm going to look at it on Saturday and Sunday. Generally, I look at it one time during the weekend, but I don't do it on both Saturday and Sunday. Any question about any of that? All right. So all of our future conversation will be in the unknown notes pages. And let me see if I can show you an unknown notes page to a previous student. I'm going to shut this down because I don't want to show private information, but I can hopefully show you an unknown notes page, what it looks like in a previous term. That should work. Unknown notes. And then I'll just get one which has some information, which looks pretty good. Oh, these are graded. I shouldn't show that. So what can I do to hide that? If I go there, oh, that gets rid of it. Okay, here we go. So let me share the screen again. Here's an unknown notes page for a previous student, and hopefully I'm not sharing that. Come on. There we go. And I guess I am sharing that, but there, I got it hidden now. All right. So you don't really need to put the date on it because the system will date it and the time, but here he's reviewing the results I sent to him, and then he's discussing what the results were for the gelatin hydrolysis test and probably the gram stain. And there's the results of the gelatin hydrolysis test, and there's the results for the PR lactose test. And he's got it right here. No evidence of fermentation because the broth remained red. And then he wrote up on 8/4, which I guess that's the same day. He wanted to do the test on oxidase. And this is not quite the best results because you should have in here the species under consideration. And later down, he's got it. He says, this eliminated Salmonella typhimurium. And then my options are narrowed down to Proteus mirabilis and Proteus vulgaris. And so then he's asking the indole test. And he doesn't state it, but you should state that an indole test will separate out Proteus mirabilis from Proteus vulgaris. And then I sent him the results, which it doesn't look like he's showing that here. But he's got the results in here that the indole test came back bright red, which was negative. No, the negative was yellow. So the indole test was positive for the indole test. And that means that Proteus vulgaris was positive. So this is what he thinks his unknown bacteria was, Proteus vulgaris. You got an M on there. That should be Proteus vulgaris. Any question about that? Let me state that on these unknown notes page, there's a, what do they call that? A dialogue area on the right here. And I'm going to ask you not to put any of your comments in the comment area. I want you to put your comments in your document. You can put them on the bottom of the document or the top of the document, and I will find them. Do not put them in the comment. If you put them in the comment, I'll just delete them because the comment is for me to give you information, and I will put the comments in. I won't put anything in the document. I don't even think I can add a document to your unknown notes page, but I can add a comment. And so I'll send everything in the comments. So don't put things in the comments because it's a real narrow section. And if you have a whole bunch of stuff in the comment section, you're going to have to scroll all the way through it to get it and try and read it. Any question about any of that? All right, let me close this.
I just wanted to clarify, are we going to be submitting a Word document every single time we have the conversation, or is there going to be like a comment section? I'm still a little confused.
Once you start working with the unknown notes page, I think it'll be obvious. You generally submit a document like a Word document, but it does accept a docx, a rich text format, a text document, a PDF file, and it does accept an HTML file. So it doesn't have to be Microsoft Word. If you do not have Microsoft Word and you want to use a word processor, save it as a rich text formatted document or a text document or a PDF file or even an HTML file. It looks like you can submit the document in the text directly. Let me check on that. Yeah, I've got it set up so you can go to the document and just start typing, and then it'll do it in text, and then you can save it and it'll be a text document. So you don't even have to make it a document and upload it. You can simply go to the unknown notes and start typing, and it'll be text entry. Okay? But most
I
people
have,
use Microsoft Word and then submit the document, and that's fine. Like I said, if you don't have Microsoft Word, just save it as a rich text formatted document or a text document or a PDF file document. There was
so I
a,
had a question. So,
uh-huh.
is the unknown list the one that's supposed to help us narrow down to get our bacteria, right?
I think so. Let me see what it's called. Oops. Just a minute. Gotta get that out of the way. I don't think I need that anymore, but let's back up. No, there's no backup on that one. Yeah, the unknown list shows you the number of species that are under consideration. And let me not do it that way. Go here. It'll be easier to see. There it is. So on the left are all of the species under consideration. The top one, there's about 13 of them, are gram-negative bacilli. And then the bottom, about 13 of them are gram-positive cocci. So these are all the species under consideration. There's about 23 of them. And these are most of the tests you can run on the top up here. And this table gives you the results for each species for each test. So for the gram test, these are gram negative. For the gelatin, let me blow this up. Gelatin hydrolysis. These species are negative. These species are positive. That's negative, that's positive, etc. Any
So
questions?
those are like the results,
Say
basically.
again? Say
Are
again?
those the results of the gelatin
I can't
for those
understand a word
species?
you're saying.
So basically for those species, you're saying that in the gelatin, the negative ones are um, the results of the gelatin test.
I don't understand your question. The negative ones are ones that do not have gelatin hydrolysis. So if you were to run the gelatin test, the gelatin would remain solid. If the bacteria has gelatin hydrolysis, then they'll be positive in the gelatin test and the gelatin will turn liquid.
Okay,
And you
thank
can
you. That
find
answers
out
my
about
question.
each of those tests, more information, by coming here. Let me see if I've got that on. Hopefully I do. Yeah, I do. So here are all the tests you can run, and it'll tell you more about each of the tests. And you should read the test before you decide to run it. And certainly after you ask it, you should read about the test. So this tells you the steps that are used in doing the test, what the test is about on the top, what the results can be. the methods you use to run the test, and then the results of the test. Okay. What's important about this is it tells you how long the test will run. And the gelatin hydrolysis test, or the gelatin test, takes at least 7 days of incubation from once you start the test before you get the results. So if you want to do the gelatin hydrolysis test, it will run for a week before you will get the results. And when you request a test, I will set it up for you and then send you the results, and you will not get the results until after the incubation time. Most of the tests take 2 days. So there's a large number of tests here you could run. Most of them take 2 days to run. The gelatin test takes the longest. It takes 7 days. The MR test is the next longest. It takes, methyl red is MR, it takes 5 days. Most of the others take 2 days. There are a few that take 1 day or less. And that would be the pigment test, the oxidase test, the catalase test. For the oxidase and the, actually just the oxidase test, if you don't have fresh growth, that will have to be grown. So you cannot get it in a day, because you'll get it in 2 days, because 1 day you have to grow your bacteria to get fresh growth, and then you run the oxidase test. But if you have fresh growth, you can get the oxidase test in 1 day, and the pigment test you can get in 1 day. The carbohydrate test takes, I think it's 17 to 28 hours. Is that right? I might have that time a little off, but it's either a little less than 1 day to 1 day. It can run for 2 days. Well, that's really slow at loading. But it's best if you run it for about 24 hours or 1 day.
And just to clarify, I know you mentioned it, but I forgot what you had said. We can only request 1 test at a time after the 3 initial ones, correct?
I haven't talked about that yet. I discussed it maybe earlier in the term, but I haven't talked about that today. Not yet. We will get to that when I'm going through the directions.
Okay.
Okay. All right, I think I'm done there and that. Let's go back to the directions. Anyways, you will need to look at all of these pages before you write up your unknown report. And while you're doing the project, you should probably look at the unknown instructions and the unknown species list. Obviously, you use the unknown notes page while you're doing it. And then every once in a while, you'll have to look at the building a dichotomous game plan. You don't need to look at that right away, but you will need to look at it at least once when you're doing your unknown project. And it probably would be a good idea to look at that one, this one, this one, and this one before you submit your unknown report at the end. Okay, and I can tell you right now, I don't have the submission button for the unknown report here, but it's going to be due March 4th, 1 day after the unknown notes pages are due. The unknown notes page, let me say, is not only a place where we, meaning you guys and I, communicate with each other for this project. It's also a place where you will be graded. This is one of your lab submissions. So this is worth 5 points. And so if you do a good job, you'll get 5 points on the unknown notes page. Sometimes students ask for the last test and then they don't bother putting the results and conclusions up. in which case they do not get all of the 5 points. They don't get more than 1 point missing, but they at least get something docked because they don't have all of the unknown notes page information up. So the pages are used for the unknown projects. And then I'm stating that if you get confused, please ask about it and I'll try and straighten out the confusion. All of your records should be put in your unknown notes page. Okay, that includes the information I will be emailing to you. So you should put the unknown number in your unknown notes page. And then you should put the first 3 tests when you start them and when you get the results. They should be put in your unknown notes page. And then when I send you the results, you don't have to copy my information. You could just say the color of the bacteria were pinkish. So that's the results. The observation is the results. And so my interpretation or conclusion is that it's gram-negative bacteria. And that would be your conclusion. So the results are your observations. The conclusions are your interpretations of the results. And as long as you have clear observations and clear interpretations, you will get the points for that. As long as you have all your results up here, you'll get all the points for your results. All of your conclusions up there, you'll get all the points for the conclusions.
Is this the one you were saying where you can enter it as a text entry?
Yes, you can do it as a text entry.
Okay.
If you want to be formal on your unknown notes page, the unknown notes page can look very similar to your unknown report that you are submitting. And some students like to do it that way because then they can just take their unknown notes page and then easily transfer it into your unknown report. But you do not have to be formal in your unknown notes page. You should be formal in your unknown report. Okay. There are some things that the unknown notes page does not have to have and your unknown report has to have. And I'll talk about those when I talk about the unknown report. Like you have to have a table in there talking about how you found your species and why you think it's that one species you think it is. And you should have references in your unknown report. And you should have a paragraph talking about the bacteria that you think your unknown bacteria is. And it just has to be one paragraph talking a little bit about the bacteria. Okay, the unknown notes page doesn't need any of that. All right, that's it. It looks like I haven't requested that you put the species under consideration in the unknown notes page. I thought I did, but it doesn't look like I did. You should have the species under consideration in your unknown report. Okay, so initially at the very start, you're going to have all the species. And then one of the early tests you're going to do is going to be the gram stain. And so that will reduce the number of species under consideration down to 50% of them. And then you'll have other tests that I will give you in the first email, and it will narrow it down even further. All right, any question about any of that? So that's how you do the unknown project. Now let's talk about the directions for the unknown project. So this needs to be updated. It's not 100 points anymore. It's 75 points. It's a 75-point lab project. And that is on the syllabus. So it's also, I think, I should check on that to make certain, but I think on the instructions I gave you, it'll tell you it's a 75 point thing. I'm using my own copy, which is probably an older copy. And yeah, it's 75 point project. I'm just using my instructions because it's a little easier running from my computer. All right, you can read about the introduction and the rationale. Let me talk about the project format. You don't really have to isolate the bacteria. When you went into the lab, I would give you a culture that had 2 bacteria species in it, and you'd have to streak it out and get a pure culture. We decided in the online course that we're just going to give you one unknown number. And so you're going to be given a pure culture from the start. But during the first week, you can say in your report, I am pretending to grow my bacteria. Okay. You don't really need to, but you can because the sample report has that in there. You have to identify your species of bacteria. Now, I gave you a spreadsheet for helping you, but you can use other media for help in identifying the species of bacteria. You can use your lab textbook, which is the lab manuals. You can use your class textbook. You can also use Berge's manual, which Berge's manual is how the spreadsheet that I gave you is based on. You should use dichotomous logic for this project. Dichotomous logic means that you ask a question, is my unknown gram positive or gram negative? Meaning, is it yes or no or true or false? And whatever the result is, you narrow it down to one of 2 possibilities. That's what dichotomous logic is. All of the tests you ask should follow dichotomous logic. That's opposed to where you were to run all of the tests on one day. Like you say, I want to do all of the possible tests, 6.5% sodium chloride, 7% sodium chloride, urease test, nitrate test, catalase test, PR glucose, PR lactose, PR sucrose, PR mannitol, PR inulin, and growth on EMB. I want to run all of these tests today. So let's start them today. That would be shotgun logic. Okay. Dichotomous logic means you'll be separating out your possibilities one test at a time. Like you do the gram test and it's gram negative. That means when you do the first test, you've now eliminated all of the gram positive species. And so all of the species under consideration are now only the gram negative. And I told you you're going to be doing the gelatin hydrolysis test. If it is a positive result, then it's going to be one of these 3 or this one of the gram positives. All of these gelatin hydrolysis negatives have now been eliminated. So in this case, I told you you're first doing the gram stain and then doing the gelatin hydrolysis. And now we've narrowed the species under consideration down to 4. Okay. Any questions about that? I guess I should go gram stain. That's not exactly how you're doing it. You're going to do the gram gelatin hydrolysis test first, and then the gram stain second, I think. But it still results on the same 4 species. Any questions about that? That's doing dichotomous logic. You'll do the PR lactose test, but actually all of them will be negative in this case, so it doesn't help you narrow it. And then you can do another test to narrow these 4. You would not want to do 6.5% sodium chloride because all of these are negative. It won't help you narrow it. You wouldn't want to do 7% sodium chloride either. You could do urease. You'll have 3 positive and 1 negative. You wouldn't want to do nitrate. You wouldn't want to do catalase. You probably wouldn't want to do PR glucose. You could do PR sucrose because then you can separate 1 from 3. I'm trying to look at a better one. The best ones would be one. Here's a good one. Nope, that's not the right one. Sorry. Let me come on over here. Here's a good one. Yes, this would be a good one. If you did the indole text next and it is positive, you narrow it from 4 down to 2. And so you would remove this one and this one from consideration. Okay, and that's kind of how you do it. You go through the table, see what your results are, figure out what is what, and then, I don't know, highlight them or just remember which one are under consideration. Now we've narrowed it down to 2, and we removed half of them under consideration. Let me state that when you're looking at this table, I think it's obvious what positive and negative are, meaning the test is positive, the test is negative. What's the V mean? A V means that the results could be variable. That means, in this case, if we had something that was positive, we'd have to take those, and we have to include all of these variables because the variable could be positive, but the variable could be negative. So we cannot rule these out. Any questions about a variable result? Okay, and it does explain on the table at the bottom what each of these means, and there's some information there. The not determined in D means treat it like a variable. We don't have the information on the not determined because the nitrate test is not run on gram-positive cocci, at least these, and so we don't have the information on what the results would be, and these tests are not normally run. So you have to treat these as variable. Okay. AG means acid and gas. A down here means acid alone, no gas production. This A parentheses G means that, yes, we have acid. Most of them have gas, but a few of them do not have gas. And I'll tell you that for our species, all of them have gas if they are A parentheses G parentheses. So you can treat that as AG for us. I think that's all of it. And then I guess the only other thing I need to say is the asterisk here, and that is these are bacilli. However, when you're looking at them, they may look like Cuccobacillus. So E. coli and E. aerogenes, and well, we'll talk about that one later, are bacilli, but sometimes they can look like Cuccobacillus. And then the same with Lactococcus lactis. It is a coccus, but sometimes they can look like a Cuccobacillus. And it kind of depends on when you're looking at the culture, as well as which media you're growing them in. Let me see if I can remember. I don't remember for Lactococcus lactis, but I do remember for Enterobacter aerogenes and Enterobacter cloaceae. If it's a freshly growing solution, meaning we're growing the bacteria in a liquid culture and it's freshly growing, meaning it hasn't been sitting there for a while, the bacilli will take on the shape of a coccobacillus. However, most solid media, the bacteria grow a little slower and they tend to be more of a bacilli, especially if the culture is not freshly grown, meaning it's 2 days old or older. It'll take on the shape of a bacillus. One other thing I should talk about, and that is this tells you a little bit about the information on the test. You really should get this information from the test itself, meaning I've got it up on the test. Oops, wrong one. Close that down. So come down here, lab, unknown tests. So you should look at that there. And let's take a look at the gram stain. Gram stain, cell shape, and growth arrangement. Come on, load. I'll tell you what it is. With the shape of the gram stain, the 3 main shapes are possible. The main arrangements are diplo, staphylo, strepto, and where is it saying here? It is difficult to determine if cocci are in clusters or chain when viewed under the gram stain. And that's because the chains can be broken apart during the production of a smeared cell. And if the bacteria are grown on a solid culture, the bacteria will clump together. So do not rely, oops, that's not spelled right, on the growth arrangement from the gram stain. To do the gram stain correctly, you should do the test, which is called the cellular growth arrangement. Cellular growth arrangement. And what you do here is you grow the cultures in liquid culture, and then you're more likely to see the cellular growth arrangement. So the cellular growth arrangement cannot generally be determined from the gram stain. That's because chains may be broken apart and cells tend to be brought together to form clumps, especially if they're grown on solid culture. To assay for this, you take special procedure, grow your bacteria in a liquid culture, and then take a loop of the liquid culture, put it on a slide, and then very gently stain the cells. Okay? So that you don't disturb the cellular growth arrangement. And then you look at the cells. So there's that doesn't look like a diplococci to me. That looks like a streptococci. There's streptococci. Blow that up. Well, that doesn't look like a diplococci. There's a diplococci right there. And there's another one right there. There's one there too. But where the pointer is, that looks like a streptococci. All right. Any questions about any of that? So let's go back to our directions. The point is you should use dichotomous logic. If you're using dichotomous logic, you're only requesting one test each time. And what you can do is you can request like a test that you can get the results in one day. You can request that. And then the next day, you can request the longer test, the test that takes 2 days or longer to run. If you use shotgun logic, I will warn you that you're not using dichotomous logic and the project should be using dichotomous logic. But if you want, please confirm and I will give you the test results that you're requesting. And if you confirm it, I will give you the test results you request, but then you will be penalized because you're not using dichotomous logic. When you write up your report, you should write up the daily lab notes. So each day you have in your unknown notes a page where you have something on the page. It may be results, it may be a test request, or it could be all of that. Okay. And then you should probably have also your interpretations of the test results. You should put that in your lab report. So document all your activities, and in the lab report for each day, you should have the day that happened. Let me state again that that's not 100 points, it's 75 points. Submit a typewritten hard copy. It's got to be that because you're doing it online. The report should use 12-point font. If you have less than 12-point font, I'm going to have a hard time reading it. So please have 12-point font. I don't really care if you double space or not. You can. I'm okay if you don't use bullet points, but you should have text in the report. You should have a description of the test you run. And if you're running the gram stain, all you need to do is say, I ran the gram stain. You do not need to tell me the steps you used to do the gram stain. I know the steps you use to do the gram stain. So you don't need to put any of that information in your lab report. You should not omit data that makes your project look better. You should include all the data you asked for and all the data you got. Even if you learn later that, I don't know, this didn't help you, or you went in the wrong direction, and you later got your mistake, and then you went back and you did it, you still have to write up the fact that you did that data, that you did that test and you got that data. You do not have to include drawings, photos, and most tables in your lab report. However, drawings, photos, and tables are very helpful. The drawings, photos, and tables must be completely labeled. Like if you got them from the internet, you should say where you got them. Words and thoughts you use in this report should be your own. If ever you use somebody else's words, and I'm talking about 3 words from somebody else, you should put it in quotes and then state where you got it from. Otherwise, it is plagiarism. And plagiarism is really important in school. I don't know if I ever mentioned that. Plagiarism is extremely important in real life too. There once was a sitting senator, I think he was from Nebraska, and some paper looked up what he did like 20 years earlier before he was a senator. And it turned out when he was writing up it was either his thesis or his dissertation, he plagiarized somebody else. And he had to resign his seat in Congress because he plagiarized. And we are that strict in college. If you plagiarize in your lab report, you'll get a 0 in it. And you don't want 0 out of 75 points. So don't plagiarize. All you have to do is put it in quotes and then say where you got it from. It is important, in fact, it's mandatory that you have a table outlining the process you use to identify your unknown organism. If you don't want to do it in a table, you can do it in a, what do you call that, a chart or a graph. And I got an example of that here. So these were all of the possible species at the start. The gram-positive coccus. Actually, that's not all the ones at the start. This is after we did the gram stain. And then they did the catalase test that narrowed it down to these 4 possible species. Then they did the PR mannitol test, and it was negative, and it narrowed it down to these 3 species. We did the PR glucose test, and the results were acid, so it narrowed it down to these 2 possible species. And then we did the citrate test, and it was positive, and that narrowed it down to Micrococcus varians. Okay, so you do need a table or a flow chart or a diagram showing how you got this species, what test you ran, and what the results were from that test. You must have that in the report. You don't need that in the unknown notes. All right, where was I? You do have to include a paragraph talking about your unknown organism. I can show you that later. So you want a paragraph talking about Micrococcus varians. And that can be many different things, but usually people talk about general things like where you can find Micrococcus varians, what human diseases it can cause, and something else about Micrococcus varians. Because you're talking about a paragraph about that organism, you should have at least one reference. And so you have to have references, and it would be the literature cited page. You can use whatever format of the reference you want, CSE, APA, MLA, doesn't matter. The paragraph talking about your microorganism should not be more than 2 paragraphs in length. So don't make it like 5 pages long. And certainly it should not exceed 1 type page for that. I think the report should be something like 5 or 6 pages long. But let me look on that. I've got that in there someplace. Not finding how long it can be, but don't make it 10 pages long. It should be something like 5 or 6 pages long. And that's including, it's not including pictures and your references. So if you're a little longer than that because of pictures and references, that's fine. You should have at least 5 pages of text. Okay. If it's less than 4.5 pages, you'll get a penalty on that. The grading criteria, let me talk about that. Will be graded on dichotomous logic. You should have the completeness of the test, what tests were ran, why you ran those tests, what the possible conclusions were, meaning what species were under consideration and what the test would show, what conclusions you drew from the test. So the results should be in your lab report as well as the interpretation, and that's what we mean by conclusion. How well the paper is organized, its clarity, its neatness will count. If it's not neat, if it's not organized, you'll get a deduction. If you successfully identify your unknown bacteria species, it will be worth some points, but not a lot. And let me go down here to see what it is. It's not here. Let me look on the web. Probably should have just backed up. Why is that so big? I think it's worth about 5 points, but it might be more. There it is. So if you get the correct species, nope, it's worth 10 points. It'll be worth 10 points if it is a species within the same group, something like E. coli and E. aerogenes, it would be worth 7 points. If it's outside the group, but the gram stain is correct, you'll get 5 points. If on the other hand, you say it's gram negative and I told you it was, gave you a gram positive, you'll get 0 points on the species identification. Where else to grade? Guess I'm gonna have to go to the grade sheet. So you will be graded on the utilization of resources. If you ran 1 extra test instead of getting 10 out of the utilization of resources, you'll get 8. If on the other hand, you had 2 tests that you did not need, then you would get 6 points out of 10, and etc. Very rarely do students, actually most students get a 10 out of this. Very rarely do they have an extra test. Sometimes students will go down the wrong road and they're just confused and they're asking for the wrong test and they don't discover it right away. In which case, very, very rarely I've had 2 students. Have I had a student who was missing two tests and they would only get 6 out of 10. Was dichotomous logic used in selecting your tests? If there were no excess tests, you'll get 10, meaning dichotomous logic was used. If you ever asked for 1 excess test, meaning 2 tests at one time, you would get 8 points. If you asked for a whole bunch of tests at one time, you'd get -8 or only 2 out of the 10. And then if you had no logic used in selecting the tests, you get 0 out of it. Were all of the relevant tests used, meaning did you omit any tests? And if no tests were omitted, you'll get 10. Most students get 10. Occasionally, I have a student who does not finish the report and they will be lacking 1 test or more. And so they will get a deduction. If they're missing 1 of the tests that they had to require the identification of their unknown, they'll get 8 points. If it's 2 to 3 tests that are missing, they'll get 6 points, and etc. Effective and clear communication. This is a region that students seem to have trouble with sometimes, and that is they'll have a few errors in your lab report. And if they're minor errors, but I can understand the logic, it'll just be a 2-point deduction. If you have a number of errors, but I still can understand the logic, what you're trying to do, it'll be -4 points. If you have some errors and I don't understand your logic, meaning there's so many errors and they're so big errors that I can't follow your logic, what you're doing, you will get 7 out of 15. And I've never had this happen, but somebody turns in a paper with lots of errors. It's hard to read. The logic, there's no logic for the test they did. Then they'd get 1 point out of 15. I've already talked about the species identification. That's a total score of 75. Okay. Let me mention that if you're missing some areas in the lab report, like you don't have your paragraph for describing your bacteria species, or you don't have your table for detailing how you identified one species as your species, then you'll be marked down for something completeness of the report. Okay. If you have a whole bunch of things missing, you might be marked down on more. I'd give you a 2 here and then mark you down somewhere else, but I've never had that happen. All right, any questions about the report or the grading of it? As I stated, you will want to think about it, take a look at the table, what the possibilities are, and it's probably easiest if you start selecting what your possibilities are, and then figure out what test you want to run. What else do I want to talk about? Oh, I've got an example of what the report looks like. Let me close that down. So this is an example of what a report will look like. This is building a dichotomous game plan. If you're not certain of what a dichotomous game plan is, you can read this and it'll go through an example of building a dichotomous game plan. Like I said, essentially what you want to do is separate out the possibilities by a test and divide it into one of 2 possibilities. So that way you're removing a large number of possibilities when the results come in. And essentially what you're going to be doing is asking for 1 test per day, or 1 test at a time. If you ask for more, you're not following dichotomous logic.
So when you said that you were going to run 3 tests, we can't run any tests until you do those 3. Is that right?
That's correct. You can't run any tests until I run those 3. And when I'm doing those 3, I'm going to set up the gelatin hydrolysis first. That will take a week to run. And then on Sunday, I will have you do the gram stain on your unknown. And you are not going to look at the results. You will only make the slide. And then on Monday, you'll do the PR lactose test. And you'll get all the results in one email on Tuesday. But you will start the gelatin hydrolysis first, then the gram stain, and then the PR lactose test. So it will be following dichotomous logic. The difference is that you're going to get all of the results to those 3 tests at one time. And that's just to make my life simpler. I don't have to send out
Thank you.
2 emails. But here's a dichotomous game plan, possible species A, B, C, and D. And then you split it out into rectangular or a piece of paper, and then it narrows it to species A and D. And then you split it by color to not white and white. If it's not white, it's species A. If it's white, species B. Anyways, that's the dichotomous game plan. I've already talked about the unknown list. Those are the species of bacteria under consideration and the possible tests you can run and the results for those tests. I've talked about that. I've talked about this. Now let's take a look at the sample journal. This sample journal was actually written by a professor way back in 2004. Travis Kubota used to teach microbiology, and he wrote up this unknown report. It's going to be a little different than yours because this is when you came into the lab. And so the first work he's going to be doing is he's going to streak out his bacteria because he's got a culture containing 2 bacteria species. I'm going to send you a peer culture, a number for a peer culture. So what he did was he streaked out his bacteria. You can see there's 2 different species here. And then he streaked out this colony and maybe that colony, I don't know. And then he grew them up, streaked them out again to make sure he got a pure culture, at least 95% or greater. And then he started doing some characteristics. One of the first characteristics he did was he noted that species B was pigmented. So he got the pigmentation results. He then grew them on EMB plates, and he looked at the growth on EMB. That would be the results. No lactose fermentation occurring on EMB. Because they grew on EMB, they should be gram negative. So that must be a different colony than this one here. And then he grew his stock cultures, which you're just going to assume you have. You can write it up in your report that you wrote a stock culture. And then he requested the gram stain. So we began the gram stain. And then that's the activity for February 4th. And it looks like he has the results on February 5th for the gram stain. And it looks like this one is gram positive cockeye. And I got to blow that up to be certain it's cockeye, but oops, that didn't blow up. It looks like it's cockeye to me. Now shrink it down. And you'll notice he's got the results in 3 different areas on the day. He's got the observations. So on a previous day, he requested a test, and then you'd put the test results under the observations. He's then got the activities for this day that he wants to run on this day. And it looks like on this day, he notes that, why isn't that a result? Oh, he's going to request the hydrogen peroxide test, meaning he's going to run the catalase test. So that's the activity he did. And then he gave the results or conclusions to the catalase test. The catalase test is one where you can get the results in less than a day. And he was in class. And so he ran the catalase test in class. And on that day, he got the results. And he said that catalase B is colony B is catalase positive. Okay. And this is a good way to separate out your lab report, where on one day you give the date, and then you separate it into observations on the tests that you requested earlier and you got the results to. And he could have put here observations on the catalase test. And he doesn't have that. The observation would be in the catalase test, I saw that there were bubbles forming when I added hydrogen peroxide to my cells. And the conclusion from that test is that this colony is catalase positive, meaning positive for catalase. Okay. All right, I'm not sure why he didn't ask for a test after that, but on the next day, he did, February 6th. He doesn't have any results or observations because he reported those on the day he ran it. Most of the time, you will ask for a test on this day, and then the next time you see it, you will give the observations on the next time you open your report to write in the unknown notes. And then here on the activities, he's looking at what test to run next. I don't know why he's doing the catalase test again. It looks like he's running the 7% sodium chloride test. So that would be the next test he's running. And then the next time he did it on February 11th, sorry, he has the hydrogen peroxide test. No, that was earlier. He gave that earlier. Let me see. What was the test he ran? 7% sodium chloride. I don't see the results for the 7% sodium chloride. Maybe he didn't give the results in this because he wanted it to grow for 2 days or longer. So let's go 2 days. That's a problem. All right, there's the activity ran on that day. Dang, 7% sodium chloride becomes available. Oh, he wasn't able to run that test because there were no 7% sodium chloride plates. That's why there's no observation for that test. And it looks like he may have identified it, so he may have never ran that 7% sodium chloride test. Anyways, do you see how to write it up? Particularly at the start, it'll be different because you do not have to streak out your bacteria and get it a pure culture. All right, if there's no further questions, I think I'm done. I will try and work on the numbers and send that out tonight sometime after the lab. It probably won't be for an hour or so, but I will work on that. All right, any questions? All right, if
Okay,
not, I'm
thank you.
done. All right, I'll see you.